So let's start with DNA structure stability and something called the worm-like chain model. So the most important thing when it comes to modeling DNA is understanding its structure and its stability. So we're going to have to go through some of its structural properties, as well as some of the energetics of DNA, including its stiffness. And then that's when we can move on to defining the worm-like chain model. So first, a quick comparison between DNA and proteins. DNA provides instructions to create proteins. So DNA is going to help to translate between polymer languages. So here's a comparison between nucleic acids and proteins. In terms of the alphabet, which is basically the building blocks, you have nucleotides for nucleic acids and you have amino acids for proteins. The words, or what is most important about each of these macromolecules for nucleic acids, you have codons, and then for proteins, you have the alpha helix and the beta strand. So those are essentially the step-ups from the building blocks to create certain combinations that will allow for further functionality. So the next step for nucleic acids are these sentences. So you create gene sequences, which are going to allow you to be able to know how to make proteins, such as these. So proteins are the robots or sub-robots that carry out functions. So the other thing that's important to understand with DNA is that it has electrostatics. It's negatively charged and is approximately given as negative E over 1.7 angstrom. DNA is going to be packaged by multivalent cations with 2+ or 3+ charges because it's so negatively charged. So this means it can form toroidal bundles, and in salty solutions with a 3+ charge, for example, DNA strands can attract each other. So viral DNA may be packed similarly, but once you move on to molecular biology courses, you should be able to learn quite a bit more about the differences between these types of DNA. So specifically, we want to focus on eukaryotic DNA for this course. Eukaryotic or human DNA uses electrostatics to pack itself. So DNA is given in red, and it's negatively charged. So just imagine a whole bunch of negative charges all around here. Then you have something called histone tails, which are in yellow, so these little wiggly things coming out, which are positively charged. So in terms of what's happening here is that your histone tails are going to be stabilized by 200 positive charges on the histone. And so the histone tails are going to be stabilized within the DNA coils by over 200 positive charges from the histone. So if I were to ask you a question about how does DNA wrap around a histone, then the correct answer would be that both DNA and histones are almost all neutral. The direct electrostatic interaction is very weak, but when they're close together, their backbone charges start to neutralize each other. So why is that? The main driving effect is the entropic gain of released counter ions, not energy. So we're going to take a detailed look into this in a bit to explain the counter ion effect. But when you take a look at the histone or the nucleosomes in general, they're almost neutral. Individually, they're going to be positively or negatively charged, but when they're close, their backbone charges are going to neutralize each other so that you form almost a neutral object. So the other thing I want to talk about is a comparison of DNA to a garden hose, or basically a coil-like substance. So you have coil-like DNA given here, and that's going to be a zoomed out view of DNA. If I zoom into any segment in here, we go back to a term that we've learned before, something called persistence length, or persistent length. So if I take a look here, which is a section of this coil-like DNA, it's locally stiff. So individual pieces are locally stiff, and we can say that DNA chain is locally stiff and almost linear when there's minimal curvature. So that makes sense. But at large scale lengths, specifically for DNA, anything past 500 angstrom, it tends to coil up. So this allows for wider conformational space to increase entropy. It's going to resist stretching or confinement. And essentially, this LP is called the persistent length, the length of DNA that's considered completely linear. Anything past this persistent length is going to be considered coil-like. So that's a key difference between the two here. Next, we have DNA being considered a twisted ladder. So we've seen this before. It's a DNA double helical structure where you have your GC pairs with 3 hydrogen bonds and your AT pairs with 2 hydrogen bonds. So that's just another way of looking at DNA and how it's formed. Base pairing is like key and lock binding. In terms of modeling, these are two general ways of modeling DNA. You have the ball and stick model and something called the space filling model. So these are more relevant to molecular dynamic simulations, which we won't go through in this course. Next, there are 3 well-known DNA helices, depending on where those hydrogen bonds take place and how many base pairs are in one rung of this helix. So you have the A form, the B form, and the Z form. Also something you'll learn more in a bio-focused course. But for us, the key differences between the 3 of them are the number of phosphates per helical turn. In the A form, there's 11 phosphates. In the B form, there's 10, and then the Z form, there's 12. So when we're creating models, we can use those parameters for our different models. So hydrogen bonding and stacking. What does this mean? So hydrogen bonding and stacking allows DNA to keep its shape and its form. So you have your hydrogen bonds between your bases, and then you have stacking between your phosphates. So this would be stacking, and between this, sideways, would be hydrogen bonding. So we've kind of talked about persistence length of DNA. We've also talked about hydrogen bonding and stacking, how it basically stacks more and more nucleotides on top of each other. So the next question leads us to where DNA is going to bend. So we can technically find and calculate the energy penalties, because as it bends more and more, maybe there's going to be some steric interactions or unfavorable interactions as certain ends, such as these two, get closer and closer together. So at short lengths, free energy is going to win and DNA remains straight. But at large length scales, DNA tends to coil up. So what does this mean? We have to take a look at some linking energetics and stiffness. So the question is, can base pairing and energetics be translated into chain stiffness or the persistence length LP? And something we can use to help answer that question is the worm-like chain model. And we're going to explain that later, but essentially what's happening is you're going to use the worm-like chain model and take a look at how it's going to affect the persistence length, and that will then tell you whether that DNA is going to bend or not. So we've talked about bending. We've kind of done a brief introduction to a worm-like chain model, just barely. The last thing we have to talk about before we actually touch the worm-like chain model is DNA denaturation. So DNA thermal denaturation involves heating up your native DNA so that it becomes denatured. So you've heard about this before. Denatured DNA is when your DNA becomes completely unwound into single-strand coils. So it can be localized denaturation or it can be general denaturation. So the melting temperature, keep in mind this Tm is now different. It's no longer a midpoint temperature, it's melting temperature. So remember we had the midpoint temperature for fractional helicity. Now we have melting temperature, which is temperature related specifically to DNA stability. So the zipper model, as you might have heard before, is where DNA denatures from one end, and there's a transition at the melting temperature because it becomes sharper as the number of base pairs increase. So let's take a look at what this means. DNA local denaturization. DNA can be locally denatured in various ways. The first way is with this bubble formation. So within that bubble, DNA untwisting and unwinding proteins, like any helicase, or a binding of a greasy molecule or DNA, anything that reduces the melting temperature, can allow for a local denaturation to occur, and then you have, for example, an mRNA strand being created. So this is just a quick example of local denaturation of DNA. We now want to move on to the worm-like chain model so that we can analyze all of these mathematically. So now we want to take a look at the worm-like chain model. And looking at these two diagrams, there's only two options we have. Number one, the DNA is going to be linear or rod-like. Or it's going to be bent. So with the two options we have, let's try and mimic DNA as entropic springs. So number one, we have our central axis for our DNA. So let's just call this the central axis of DNA. It's going to have a length of L. So then, the next thing we want to label are the DNA strands themselves. And there's going to be two of them. One of them is going to start here. And the other at the bottom. So now we have our two entropic springs for our DNA strands, and with any spring, you have your spring constant K. The length of each spring we can define as A naught. And we also have the radius of each of these cylinders, which we can call Z. So we have our linear rod drawn out. We can now take a look at what happens when we bend the rod. So now I have this, and the key thing here, it's no longer L. Well, I mean, sorry, it is L. So this is the L is going to change for the top and the bottom springs though. So L is the length of the central axis. So that doesn't change. But you do have this. And over here, you have L plus some sort of change in delta L. And we're going to assume that for the bottom strand, you have L minus some sort of change in delta L, because that delta L, or change in length, is going to be so small that we can only consider it as a microscopic entity. So apart from that, what we can do is calculate some sort of angle of bending. So we take a look here, and we call this our radius r. And then here, we're going to have some sort of angle theta. So just to recap, these are the two primary methods of representing DNA as two entropic springs. And for the next few slides, we're going to start analyzing different types of properties for this. We're going to be looking at what delta L is, going to be looking at how to define K, the bending energy, as well as figure out how we can tell whether a rod is going to bend or not, depending on the value of delta L compared to a persistence length. So then, from the diagram, we can see that theta is going to be equal to L plus delta L divided by R plus Z. So we're trying to use similar triangles here, and that is going to be equal to L over R. Okay, so try and take a look to see how we figure that relationship out. And once we have this equation, we have L plus delta L times R equals L times R plus Z. We can conclude that delta L R, let's see, work this all out first. We have LR plus delta LR equals to LR plus LZ. You're going to be able to cancel these two out. And you have delta L equals to LZ over R. So then, we've now defined delta L. Now we take a look at the DNA spring constant K. Okay, so we're going to take a look at that now, and let's say to find the total spring constant, we're going to say K total is going to be the spring constant of this thing that you saw before. So we had that. And we had some sort of K total associated with all of them. Actually, not K total. We had the K individual for each one of these. So if we want to figure out the total spring constant for all of these, we're going to take K individual and divide it by the number of springs. And that is going to be equal to K divided by the number of springs, which we aside number of springs would be, we had our L here, and each one of these was A naught. So that means we have L divided by A naught. So that means K total is going to be equal to K A naught divided by L. And now we have an equation to calculate not just our individual spring constants, but the total spring constant as well. So next we want to find the bending energy. So the bending energy, we're just going to say, let's define the bending energy as E bend. So I won't derive this fully, but the energy of bending is going to be 2 times 1/2 K total times delta L squared. So what does this tell us? And where is it coming from? So if you think about it, it looks familiar. It's quite Hookean, so it comes from Hooke's law. Where F is equal to negative kx. So if I'm taking a look at the energetics of this, I'm going to have U being equal to 1/2 kx squared. So x in this case is going to be the change in distance, delta L, and then k in this case is going to be K total, because we're looking at the entire spring constant of the whole rod that we're looking at. And then we have this being the E bend of one strand of DNA. So remember, each rod had two strands of DNA, so we'd have to model it as two entropic springs. So therefore we have 2 times 1/2 kx squared, or we have E bend equals to 2 times 1/2 of k total times delta L squared. So all we have to do now is just plug things in. We're going to have, let's see, k total and we just want to rewrite things like this. We have 1/2 times 2 k total delta L squared, and there's a reason why we're doing this, because we can define certain expressions as different terms. So then we have 1/2 2 k total from before was k a naught over L, and delta L we defined as LZ over R. That has to be squared. So between all of that, we can then say E bend is equal to 1/2. I'm going to rearrange some of the terms here, so just say rearrange some terms. And we get 2 k a naught z squared times L. L squared over L is just L, so it's going to be L over R squared. Okay, so some important things to note from this. Although I have my bending energy, this term here is called the microscopic entity. And this term here is called the macroscopic entity. Why is that? It's because L and R were the ones that we've measured macroscopically, whereas A naught and Z were the small changes in distance related to individual portions of the entropic spring of DNA. So between the two of these, there is something else we have to mention, and that's part of the microscopic entity. This entire term here is also considered KB, so, or not KB, kappa B. Okay, so let's put a star here. The bending modulus kappa B is defined by microscopic quantities. So we've now defined what kappa B is, and for future slides that are coming, we can then take a look at what that means. So of course, the next question is, what is kappa B, apart from knowing it's the bending modulus? So let's just say we actually want to be able to figure out how to apply kappa B to the worm-like chain model and the significance of kappa B. So I think a better title for this slide would actually be the significance of kappa B versus physical meaning of it, because we kind of define the physical meaning as the bending modulus, or how much it bends. So now we have E bend is equal to 1/2 kappa B L over R squared. But from before, we also had theta being equal to L over R. So E bend could technically be rewritten as kappa B over 2 times theta squared over L, which is equal to kappa B over 2 theta over R, depending on how you want to substitute that theta and what you want to cross out. This means E bend is going to be equal to the summation of 1/2 kappa B Li over Ri squared. So these are all interrelated. Specifically, these two can calculate the individual energies of bending. This one is going to calculate the total energy of bending across an entire chain. So let's just say this. Will be used for a worm-like chain. Why is it worm-like? Let's take a look at what this even means. Let's say you have DNA and it kind of looks like this. Okay, so I have my worm-like chain model of DNA, and there's going to be quite a few bends here. So how can I model this? I can model it with A. I have one sphere that I've drawn here. Here's another arc. I can represent that using another. Make that a little darker. This will be RB. Let's not use blue. Let's use green. I have another bend here that I want to mimic. RC. And of course you can have more if you want. These are just some examples. And I'll use black for this last one. RD. So you have all these different bends in your worm-like chain. And so if you want to calculate the energy of bending, then you would use the summation of all of the contributions coming from each of these circles. So then to define this, the worm-like chain model is going to be defined as E bend is equal to So we have kappa b over 2, but I also want the summation of Li over Ri squared. So this is technically the integral from 0 to the total length of 1 over r squared s ds for a small step size length. So that specific model is one that, once again, we won't specifically apply that for a testing situation, but we might work together on it for an assignment or a group project. So we've done quite a few calculations and derivations for different properties of the worm-like chain model for DNA. Now we go back to something familiar, persistent length or persistence length. LP is going to be the length scale with which the chain remains straight. So anything beyond that persistent length LP, the chain's going to coil up. So what we're going to look at here is delta L compared to delta L compared to LP. So I'm just going to star this. Delta L is key in determining bending or no bending. So if I want to compare delta L to LP, there's only 3 things that can happen. It can be less than, approximately equal to, or greater than LP. So let's take a look at what happens if delta L is less than LP. Remember our delta L was going to be our change in length for the entropic springs at the top and bottom. The greater the delta L, the assumption is the greater the bend we have. So in this case, if delta L is a lot smaller than LP, the assumption is that delta L delta L gets close to 0, or something that's essentially negligible. In that case, we can assume that there is no delta L, and that means the length of our rod is also going to be the length of our entropic spring. So that means we have something like this. So essentially we have this being a length of L, this being a length of L, and this also being a length of L. And we're saying that the plus and minus delta L is so small that it's much smaller than the persistence length. It's not going to have a huge effect on the bending of the rod, and therefore we can conclude that for delta L is less than LP, we have a linear rod. Now, what happens when delta L is approximately equal to LP? Conceptually, we can think of it as we now have a delta L that is significant. It's now going to be approximately equal to the persistent length, which is just enough that it almost bends. So therefore, we're going to have something called the perfect bend happening. So we have this. I know this looks like a bend already, but we have this being L. we have this being L plus delta L, and we have this being L minus delta L. Or it could be L minus LP or L plus LP. So what this is saying is that we now have something that's really close to bending, but not bending. So it's going to be considered just bending. So the other assumption here that we can make is that your R is going to be approximately equal to delta L, which is going to be approximately equal to LP. So that's one of the common assumptions that are made. We won't really go into depth with this unless you're doing it for one of your projects and want to explore this a little bit more. But that's just a general understanding of what happens when delta L is approximately equal to the persistent length. Next, we have delta L being greater than LP. So when I'm talking about just bending, and this is the perfectly circular case, where the bend is perfectly circular. When delta L is much greater than LP, we have a problem in that it's going to be very curvy. In that case, we have no idea what's happening. Could be something along the lines of this. And in this case, we just consider this, it's no longer circular or linear. The other term for this would just be coil, or coil-like. So now we have three different ways of comparing delta L to LP. Let's now take a look at being able to figure out what the persistent length is. The persistent length is going to be, let's first start off with 1/2 kappa B. We have LP over LP squared is approximately equal to kBT. So try and think of where this comes from. Let me just put this. Try and take a look at one of the previous cases for delta L compared to LP and try and figure out where we took that from and why. It might be a good question to answer. So next, once we have this, for the persistence length, it's also defined as a length that's going to be equal to kBT. So we have LP being approximately equal to kappa B over kBT. So we can say LP is equal to kappa B over kBT, and that is going to be called the persistence length. Okay, so as an aside, I just wanted to quickly mention a few things. to extend your knowledge. When we try and calculate probability, which is something we'll get into in the next portion of this unit, the probability of something bending, let's just draw probability of this, where, let's say, R is equal to delta L, is approximately E to the negative E bend over kBt. So that means E bend of this is approximately equal to kBt. So now try and think of how that relates to this in order to derive that expression we got at the beginning. So to wrap up, we just want to go through a summary of different cases we've looked at and a summary of the worm-like chain model. There are 3 cases to consider, and that is when 1, or A, when our length is less than LP. We have a rod-like or linear DNA structure. So we're going to have something that looks like this. B, if our length is approximately equal to LP, both the rod-like and curved effects are comparable. So you get a curved rod. And that perfectly curved rod is going to look like this. Next, we had the length being greater than LP, and that means you end up with an entropic coil. And an entropic coil is going to look like a coil, as one would expect. Like this. So these were the 3 cases that we considered. It'd be really good to remember these. And then we take a look at the worm-like chain model again. So we're just going to summarize everything that we've taken a look at here. For the worm-like chain model, we had some sort of chain that started here. And let's say like this. So we have a start and an end to the chain. When we want to model something like this, we can model it using a circle with a radius r. We were able to calculate the energy of bending, which is kappa b over 2, integral from 0 to L being the total length L, from 1 over r squared s ds. So that was one of the main equations. We know that LP is equal to kappa b over kBt So what are the things to know? Key points. We can predict bending configurations, by comparing delta L to LP. We were able to group and define certain microscopic or macroscopic parameters, and we were able to find the bending energy for long chains. So that summarizes it for the worm-like chain model. Next, we are going to look at DNA elasticity and then merge all of our knowledge together to try and come up with better models.